Thanks very much for the subscription. For those one who subscribed, those one who have not yet subscribed, please do one thing: subscribe, put on a notification, and we continue to learn, we continue to enjoy the true facts of medicine and health. Today we are going to look at an important pathway, and that is the mTOR pathway, mammalian target of rapamycin pathway. And this pathway is very important in regulating cell proliferation and growth, cytoskeleton formation, and it's also linked to autophagy when we have some other signaling mechanisms that substances that come and interact with this pathway. This is also linked to protein kinase pathway, as we are going to see later on in our subsequent videos. In this pathway, we are going to be looking at it. We are going to be encountering some short forms like these ones. And these ones we are going to be standing for, where we have mTOR. This one is going to be standing for mammalian target of rapamycin. Where we're going to see raptor, this one is regulatory associated protein of mTOR. Richter, this one is rapamycin. Insensitive companion of mTOR. Depta, this is going to be depth domain containing interacting protein. AKT, this one is also known as protein kinase B. PI3K is phosphorinositide. Phosphorinositide 3 kinase. Then PIP2 is phosphorinositide 4,5 phosphate. Then PIP3 is phosphorinositide 3, 4, 5 phosphate. Then 4EB is 4E binding protein. 4E binding protein. Then this one is going to be eukaryotic, eukaryocytic, not eukaryotic, it is eukaryocytic. Eukaryocytic descriptional initiation factor 4E. Then PDK1 is phosphorinositide dependent protein kinase. Protein kinase 1. So these are some of the short forms we are going to be coming across in this pathway. Let us see the difference between, because mTOR has two molecules. We have mTOR C1 and mTOR C2. So let us see the differences between these two molecules. mTOR C1, I have mTOR C2. What they have in common is the depta depta molecule, and another one is MLSTH. So these ones, they have them in common. This one also has depta and MLSTH. The difference comes in, this one has raptor molecule, this one has Richter, this one has price 40, this one has scene 1. This is mTOR C2, mTOR C1. Another difference comes in the phosphorylation of the protein kinase B. mTOR C1 phosphorates protein kinase B at the threonine residue, and this is 308, and this one phosphorylates it at this was phosphorylates at a serine residue 473. So those are some of the differences between these. So let us go to the pathway and we see what takes place in the pathway. So when we have an antigen being recognized at the TC receptor here, so this one being our antigen, in the presence of CD8 co-stimulation, what are we going to have? We are going to have phosphorylation and activation of phosphorinositide 3 kinase. So what we are going to have here, we are going to have phosphorylation and activation of phosphorinositide 3 kinase. So when this one is phosphorylated and activated, what is going to happen next? So this one is going to lead to phosphorylation of phosphorinositide 4,5-phosphate or PIP2. And this one is going to be phosphorylated to phosphorinositide 3,4,5, which is PIP3. And the enzyme involved here is phosphorinositide dependent kinase 1. So once we have PIP3 being formed, what is going to happen next? We are going to have this PIP3 binding on the protein kinase B. It's going to bind on protein kinase B, but specifically on the threonine residue in position 308. So once it binds here, it's going to activate the AKT or protein kinase B. Once this protein kinase B is activated, it's going to do 2 roles. The first role is going to be inhibition of tuberosclerosis complex 2. The first role is going to be inhibition of tuberosclerosis complex 2. When this one is inhibited, we are going to have activation and formation of mTOR raptor complex. And this complex is going to be activated by a molecule called RAS analog, which is abundant in the what? In the brain. That is REB. So we are going to have REB binding on mTOR C1, activating it, leading to formation of this is mTOR raptor complex. Once mTOR raptor complex is formed, what is going to happen next? This complex is going to phosphorylate and activate 2 proteins. One of the proteins is B70S6 kinase. It's going to. So the second protein we are going to have here is the 4E binding protein. And this protein, in the absence of the antigen here, if this pathway is not on, is not stimulated, this protein acts as an inhibitor of what eukaryotic transcription initiation factor 4E. So what are we having? We have binding 4E binding protein. And we said in the absence of a ligand, the antigen or the T cell receptor, we are going to have it acting as an inhibitor of what? Of eukaryotic transcription initiation factor 4E. But during the when the system is activated, we are going to have it. We are going to have it phosphated and activated. It's not going to inhibit the molecule. So if this molecule is not inhibited, together with the P70S6 kinase, we are going to have proliferation. We have cell division. So when the system is not on, this 4E binding protein is going to inhibit the eukaryotic transcription initiation factor 4E. So we shall not have proliferation and cell division. The same regards, if you have factors like glucose deficiency, starvation, we are going to have tuberculous factor being activated. When this one is activated, it's going to inhibit, acts as an inhibitor to mTOR C1. So if mTOR C1 is inhibited, we are going to have activation of ORC1, which is involved in autophagy. Then we are going to have the second instance where we have interleukin 2 binding to interleukin 2 receptor. If we have interleukin 2 binding here, when it binds there, what are we going to have? We are going to have formation of mTOR C2. This one is going to complex with mTOR Richter. So when this complex is formed, it's going to come and phosphorate and activate the protein kinase B. This one is going to bind specifically on the serine residue 473. So once it binds there, we are going to have regulation of cell polarity and cytoskeletal formation. Now we have a drug. Since mTOR is mammalian target of rapamycin, we have a drug called rapamycin. So rapamycin acts to inhibits both mTOR C2 and as well inhibit mTOR C1. And we are going to see in the mechanism of autophagy that inhibition of REB, which is the one responsible for activation of mTOR C1, we are going to see inhibition of mTOR C1. When you inhibit mTOR C1, we are not going to have cell proliferation and dividing. What else is going to occur? mTOR C1 acts as an inhibitor on the ULK gene. So when ULK gene, ULK gene is not inhibited, we are going to have to see autophagy coming in. We are going to see that in the mechanism of autophagy.